Hello everyone, welcome to Concept and Fact of Life Sciences. Today in this video, we are going to discuss about the functions of plasma membrane. As we already discussed that one of the important function of plasma membrane is transportation. This is the third lecture on this topic. In our previous video, we have already discussed about the type of transportation, that is active and passive, the transporters involved in that particular type of transportation, and the process of transportation. So in this video, we will take some specific type of transporters, which are the ABC class of transporters. Now these ABC transporters, these are also known as the ATP binding cassettes. So these are the ATP binding cassettes. So there is an involvement of energy. So these transporters, they remain involved in the process of active transportation. So here the transportation of molecule is against the concentrational gradient, means from their lower concentration to the higher concentration. पैसिव ट्रांसपोर्टेशन में क्या होता है, अपनी हायर कंसंट्रेशन से लोअर कंसंट्रेशन की तरफ मॉलिक्यूल ट्रेवल करता है. बट दीज एबीसी ट्रांसपोर्टर, सिंस दे आर इन्वॉल्व इन द प्रोसेस ऑफ active transportation, so here the molecule, they will get transported from their lower concentration to the higher concentration, and for this you require the investment of energy, and the energy it is invested in the form of ATP, since these are the ATP binding cassettes. Now if we talk about the structure of these ABC transporters, they are generally made up of two domains, or we can say they are made up of four domains, two types of four domains we can say. Here we are having two NBD domains. NBD means these are the nucleotide. These are the nucleotide binding domains. And again there is a presence of two TMD domains, and these TMD, they stands for the transmembrane domains. So in ABC transporters, there is a presence of two nucleotide binding domain and two transmembrane domains, as we can observe in this particular diagram. It is representing a ABC class of transporter, which is having the two transmembrane domain. These white lines, they are representing the lipid bilayer, and within this lipid bilayer there is a presence of two domains which are known as transmembrane domains, and there is a presence of two NBDs, that is nucleotide binding domain, where there is a binding of ATP. Now these ABC class of transporter, they remain present, or we can say this ABC transporter family, it is the most ancient type of family which occurs from which occurs in most primitive life form as well as in most advanced life form. Means they remain present in both bacteria as well as in case of the humans. So it is an ancient gene family or ancient class of transporters we can say. Now these ABC transporters, they have different functions in case of prokaryote and in case of eukaryotes. If we talk about the prokaryotes, they are generally involved in the process of influx of nutrients. While if we talk about the eukaryotes, they are generally involved in the efflux of toxins and drugs. So ये process देखने से पहले अपन देख लेते हैं how this ABC class of transporter it functions. So try करते हैं function को समझने की इसकी. So this structure, it is representing the different orientations of ABC class of transporters. होता क्या है, whenever the ligand it will bind to the transmembrane domain, there is a binding of two nucleotide to the nucleotide binding domain. Nucleotide ATP, it is a nucleotide, adenosine triphosphate. So there is a binding of nucleotide to the nucleotide binding domain. After this, there is a dimerization. We can say there is a dimerization in this domain. The dimerization of nucleotide binding domains, it will make conformational change in the transmembrane domain. The transmembrane domain, they changes their orientation, and there is a transportation of the molecule towards the inner side of the cell. Again, after transportation of this molecule towards the inner side, now we can say the nucleotide binding domain, now it remain attached with the ATP, because the ATP, it get hydrolyzed into the ADP. Now the binding of ADP to the nucleotide binding domain make conformational change in the NBD, which in turns make the conformational change in the transmembrane domain, and it will return into its original configuration, और फिर से अपनी original configuration में आ जाता है. Again we can say whenever there is a binding, whenever there is a binding of some ligands onto the transmembrane domain, there is a binding of we can say, then there is a binding of some nucleotide onto the NBD domains. There is a dimerization of these NBD domains, make conformational change in the TMD domains, and there is a transportation of molecule towards the inner site. After transportation, ATP get hydrolyzed into the ATP, and the ADP binding state, the NBD, it make the conformational change in the TMT, and the TMD, it will comes into its original configuration. So in this way, there is a transportation of molecule from inner side to the outer side to the inner side. So in the ABC class of transporter, we are having two transmembrane and two nucleotide binding domain which remain present toward the cytosolic phase. तो इस तरीके से कुछ ABC ट्रांसपोर्टर्स काम करते हैं. नाउ इन केस ऑफ द प्रोकैरियोट्स में क्या करते हैं ये? In prokaryotes, suppose this is a bacterial cell or the prokaryotic cell, there is a presence of ABC class of transporters on their membrane, and in these bacterial cells, they generally involve in the process of nutrient uptakes. ये nutrients के uptake में help करते हैं. There is a transportation, there is an active transportation of nutrients from outer side to the inner side of the cell. So they are generally involved in the influx, we can say. The transportation from outer phase to the inner phase, or we can say transportation from outside the cell to the into the cell, we can say. Now, in case of eukaryotes or eukaryotic cell, these ABC class of transporters, they are generally involved in the efflux. Efflux means it will transport the molecule outside the cell. तो यहाँ पे जो molecule है, it will efflux the drugs outside the cell. तो जो drug अंदर आपने दिया है, उसको क्या कर देगी ये? efflux कर देगी, उस cell से बाहर निकाल देगी. कौन? ABC class of transporter. So the function is entirely different. Mechanism वही रहेगा. जो मान लीजिए ड्रग की बात करें, the drug it will bind to the TMD domains. Then there is a binding of ATP to the nucleotide binding domain. It dimerizes. Then there is a conformational change, और जो ड्रग है उसको बाहर efflux कर दिया जाएगा, और influx के केस में अंदर की तरफ ट्रांसपोर्ट हो जाएगा. Now these ABC class of transporters, they were firstly discovered in the cancer treatment, है ना? कैंसर ट्रीटमेंट के अंदर सबसे पहले डिस्कवर किया गया था इन द प्रोसेस ऑफ मल्टीपल ड्रग रेजिस्टेंस, है ना? जो एबीसी क्लास ऑफ ट्रांसपोर्टर्स थे, दे आर क्रिएटिंग द मल्टीपल ड्रग रेजिस्टेंस इन द कैंसरस सेल. सपोज इट इज ए कैंसरस सेल, it is a cancerous cell, and you have designed a drug for this particular type of cancer, and you have targeted this drug to this cancerous cell. Now within the cancerous cell, this drug, it will be effluxed by these ABC class of transporters, and these transporters, they reduce the effectiveness of that particular type of drug. So देखा गया कि कैंसरस सेल है, वो कीमोथेरेप्यूटिक ड्रग्स को इफ्लक्स कर रहा था. ठीक है, तो पता लगाया गया कि भाई किस वजह से इफ्लक्स कर रहा है. दैट इज ड्यू टू द प्रेजेंस ऑफ दीज एबीसी ट्रांसपोर्टर्स, जिसके वजह से जितनी इफेक्टिवनेस उस ड्रग की होनी चाहिए थी उतनी इफेक्टिवनेस हो नहीं पाई. सो इन द ट्रीटमेंट, इन कैंसर ट्रीटमेंट, मल्टीपल ड्रग्स आर यूज्ड, है ना? एक ही ड्रग यूज नहीं करके वी आर यूजिंग द मल्टीपल ड्रग टू prevent the cell becoming resistant for that particular type of drug. मल्टीपल ड्रग कीमोथेरेपी में यूज किए जाते हैं जिससे कि वो सेल जो है कैंसरस सेल वो किसी पर्टिकुलर टाइप ऑफ ड्रग के लिए रेजिस्टेंट ना हो जाए। जब तक उसको पहचान करके ड्रग को इफ्लक्स किया जाएगा तब तक काफी हद तक वो अपना काम कर चुका होगा। सो दीज वी कैन से एबीसी क्लास ऑफ ट्रांसपोर्टर firstly discovered in the cancer treatment where they are showing the multiple drug resistance. Okay. Similarly, MDR, it is a multiple drug resistance. Okay. Now, another type of ABC transporters, they are called as CFTR. And what is CFTR? These are cystic fibrosis transmembrane conductance regulators. Okay, and these CFTR, they also belongs to the category of ABC transporters. They generally remain present on the lungs membrane. कोशिश करते हैं इसके फंक्शन को समझने की। CFTR, they generally remain present on the lungs membrane and they will transport, they are the transporters of chlorine. Okay, these are the transporters of chlorine जो एक्टिवली क्लोरीन को ट्रांसपोर्ट कर रहे हैं अगेंस्ट देयर कंसंट्रेशनल ग्रेडियंट। एंड द मूवमेंट ऑफ chlorine from, we can say, lung epithelial cell to the mucus layer, it will also bring the water with them. So we can say the mucus, it remains thin and slippery, which makes the respiration easy. Now, if there is a malfunctioning, suppose कीजिए जो आपका CFTR है वो काम नहीं कर रहा है, in that case protein, it will remain present on the we can say lungs membrane, but it is not functional. If it is not functional, there is no transportation of chlorine ions, and if the chlorine ion will not get transported into the mucus layer, then the water will comes back into the lung epithelial cell that make this mucus thick, and it will become we can say multiple bacterial growth will be there, and it will create a chronic condition. So the disease which is due to the malfunctioning of this CFTR, that is a ABC class of transporter, it is called as cystic fibrosis. उसे अपन नाम देते हैं cystic fibrosis. Now another type of ABC transporter, it is known as floppase. Floppase, जैसा कि अपन ने बात की when we were studying the membrane structure, वहां अपन ने देखा था कि there are membrane asymmetry, and that asymmetry is maintained by two type of ATPases. One is known as flippase, जो P type का pump था, and other is the floppase, which belongs to the category of ABC transporter. And the function of this floppase is to transport the phosphatidylcholine and the sphingomyelin toward the outer phase of plasma membrane. So this is all about the ABC transporters. Next अपन बात करते हैं, there is another type of transportation which is known as the transportation of macromolecule. This transportation is also known as the cargo transport or cargo transportation. Larger molecules का आप transportation यहाँ पे कर रहे हैं. So in other words, this type of transportation is also known as the bulk transport, है ना, इसको bulk transport भी अपन कहते हैं. Now, for transportation of small molecule, charged molecule, we have different type of transporters in the form of channels, carriers, and pumps. But for the transportation of larger molecule, we have different mechanism. Now, macromolecules, they get transported through two processes, which is known as phagocytosis and the pinocytosis. Now, the phagocytosis, it is the uptake of solid particles, while the pinocytosis, it is the uptake of liquid particles. So it is the basic difference between these two processes, which is the uptake of cargo molecule. One is phago, solid particle uptake, other is pinocytosis, that is liquid particle uptake. Now the phagocytosis and the pinocytosis, these are the specific terms. but the process which is utilized over here, it is known as receptor mediated endocytosis. Means किस तरीके से, किस मैकेनिज्म से ये ट्रांसपोर्ट होते हैं, उसे अपन कहते हैं रिसेप्टर मीडिएटेड एंडोसाइटोसिस। अभी किस तरीके से रिसेप्टर मीडिएटेड एंडोसाइटोसिस होता है, यहां देखते हैं अपन इस डायग्राम के अंदर। सो यहां अपन देख सकते हैं, वी आर हैविंग ए कार्बो मॉलिक्यूल विच इज टू बी ट्रांसपोर्टेड इनटू द सेल। सो सबसे पहले क्या होगा, this carbo molecule, it will get bind to the receptor. We are having the plasma membrane. Suppose थोड़ा पहले इसको डिस्कस कर ले, we are having the white lines which are suggesting or which are representing the bilayer, lipid bilayer it is, ये lipid bilayer है, and in this lipid bilayer we are having a receptor for that particular type of carbo molecule. Beneath the plasma membrane, we are having a layer of protein which is made up of clathrin. So there is a layer of clathrin proteins. Outside the cell, we are having a molecule which is to be transported into the cell, that is a carbo molecule, larger molecule. Now this larger molecule, it will get first bind to the receptor. The binding of carbo molecules to the receptor, it will make the conformational change in this particular receptor. This receptor in turns make the conformational change in the clathrin protein, and this clathrin proteins along with the receptor and the membrane, it will comes into the cytoplasm in the form of clathrin coated pits. समझने की कोशिश करते हैं इसको थोड़ा सा अपन यहाँ पे। सपोज okay, suppose we are having the receptor bounded with the carbo molecule. So first there is an, we can say the clathrin along with the two types of membrane, okay, outer and the inner layer of membrane, it will form this type of structure. It is having the receptor along with a carbo molecule. So this type of structure, it will form first after the binding of carbo. There is a conformational change in the receptor which makes a conformational change in the clathrin protein. It will form a pit-like structure. Now this clathrin coated pits, there is a coating of clathrin, that's why it is called as the clathrin coated pit. It will get detached from the outer membrane in the form of a, we can say, clathrin coated pit. So ये detach हो जाएगा outer membrane से in the form of a clathrin coated pit. Now for this detachment, we are having the functioning of a small class of G protein which is called as Rho GTPase. It utilizes the energy in the form of GTP. The GTP, it will get hydrolyzed into the GDP. तो क्लियर कर लेते हैं इसको. There is an hydrolysis of GTP into the GDP plus inorganic phosphate. After this, after the activity of this Rho GTPase, it will come into the cytoplasm in the form of a clathrin coated pit. Within the cytoplasm, this clathrin coated pit, it will get fused with the lysosome. And as we all know, the pH of the lysosome is around 5 due to the presence of more H plus ion due to the activity of B-type ATPases, जो अपन ने पहले वीडियो के अंदर देखा था. So there is a fusion of this lysosome with this clathrin coated pit that lead to the formation of a structure which is known as phagolysosome. तो phagolysosome जो लाइसोसोम का कंटेंट था वो कहाँ आ गया, अब आपके इस क्लैथरिन कोटेड पिट के अंदर आ गया. एंड नाउ द एनवायरनमेंट विद इन दिस इट विल बिकम एसिडिक ड्यू टू द प्रेजेंस ऑफ लाइसोसोमल एंजाइम्स, वी कैन से दैट आर द एसिड हाइड्रोलिसिस, और जो इनर जो एनवायरनमेंट है वो दैट विल बिकम द एसिडिक. नाउ ड्यू टू दिस एसिडिक एनवायरनमेंट it will make a conformational change in the receptor. Now this receptor, it is not able to recognize this cargo molecule, and it will leave the cargo molecule within the, we can say, within the cytoplasm. So the cargo molecule, it will get transported into the cell cytoplasm. After this remain phagolysosome, it will again get fused with the plasma membrane, and now we are having the receptor again toward the outer phase of the cell, है ना. ये वापस से जाकर के उसमें अटैच हो जाएगा, फ्यूज हो जाएगा और रिसेप्टर्स को बाहर की तरफ प्रेजेंट कर देगा. अगेन वी कैन से whenever there is a binding of this cargo molecule, again there is a conformational change in the, clathrin protein that lead the formation of a clathrin coated pit, fusion with the lysosome, phagolysosome, make conformational change in the receptor. It will release the cargo molecule into the cytoplasm and comes back onto the cell surface and represent the receptor again. So in this way there is a transportation which is known as bulk transport or cargo transport. ठीक है, कुछ एग्जांपल्स ले लेते हैं अपन इसके किसके, कार्गो ट्रांसपोर्टेशन के या बल्क ट्रांसपोर्टेशन के. वन ऑफ द मोस्ट इंपॉर्टेंट एग्जांपल इज द ट्रांसपोर्टेशन ऑफ कोलेस्ट्रॉल. स्पेलिंग थोड़ी सी गलत हो गई है. इट इज द ट्रांसपोर्टेशन ऑफ कोलेस्ट्रॉल. सो कोलेस्ट्रॉल ट्रांसपोर्टेशन की अगर अपन बात करें तो कोलेस्ट्रॉल, suppose it is a dietary cholesterol in the intestine, we are having the blood and we are having the cell which can utilize the cholesterol, or those cells which are having the receptor for cholesterol. Now in the dietary cholesterol, it cannot be directly dumped into the blood. अगर आप क्या करेंगे, इस कोलेस्ट्रॉल को यहां से सीधा उठा करके ब्लड में डाल दोगे तो क्या होगा, it will get we can get clumped, इकट्ठा हो जाएगा और क्या करेगा, डिफरेंट टाइप्स ऑफ कंडीशंस इट विल क्रिएट लाइक द ब्लॉकेज ऑफ योर ब्लड वेसल्स. सो आप डायरेक्टली कोलेस्ट्रॉल को ब्लड में नहीं डाल सकते बिकॉज इट विल गेट एग्रीगेटेड. सो टू ट्रांसपोर्ट द कोलेस्ट्रॉल यू फर्स्ट हैव टू make a structure which is known as chylomicrons. Chylomicrons क्या होता है, जस्ट you have what you have did कि आपने जो कोलेस्ट्रॉल है उसके चारों तरफ क्या बना दी, एक लिपोप्रोटीन लेयर. You have surrounded the cholesterol with a lipoproteinaceous layer and you have added some tags on them in the form of a apolipoproteins. और कुछ एपोलिपोप्रोटीन्स को आपने यहाँ पे ऐड कर दिया. नाउ दिस स्ट्रक्चर is known as chylomicrons. What are chylomicrons? Simply you cannot directly transport the cholesterol in the form of cholesterol because it will get aggregated. You first we can say you first embedded that particular cholesterol in the lipoprotein layer or lipoprotein bags, and this structure of lipoproteins and cholesterol it is called as chylomicrons. So इस कंडीशन में जो आप देख रहे हो, it is generally representing a LDL. Why it is a LDL? It is low density of lipoprotein क्योंकि it is containing more amount of cholesterol and less amount of lipoproteins. That's why it is called as low density lipoprotein, and it is, we know that it is a bad cholesterol. ये bad cholesterol है, ठीक है. इसकी अमाउंट कम रहनी चाहिए बॉडी के अंदर, otherwise it will create some problems. So this LDL, or we can say cholesterol, or we can say more precisely the chylomicrons. These chylomicrons, they can be we can say received by the cells which requires the cholesterol. Which requires the cholesterol. So it will bind to the cells which require cholesterol, and we are having the similar structure like there is a plasma membrane, we are having the clathrin, we are having the receptor. After binding of cholesterol to the receptor, there is, we can say, इसको टॉप पे ले लेते हैं, there is a conformational change in the clathrin layer which makes a clathrin coated pit that fuses with the lysosome to form the phagolysosome, and in this way there is a transportation of this chylomicron into that particular organ. Now, after utilization of some amount of cholesterol, if this comes back into the bloodstream, means now it is having lesser amount of cholesterol and more amount of lipoproteins. ज्यादा अमाउंट में क्या है, अब लिपोप्रोटीन्स है. नाउ इट इज नोन एज द HDL. इसे अब कहते हैं अपन HDL. So the difference between the LDL and HDL, it is a good quality cholesterol. इसे अपन कहते हैं good quality कोलेस्ट्रॉल. क्यों गुड क्वालिटी कह रहे हैं? क्योंकि इसमें कोलेस्ट्रॉल की अमाउंट ही कम है. इसमें हाई, ज्यादा डेंसिटी किसकी है? लिपिड और प्रोटीन्स की है. दे हैव लेसर अमाउंट ऑफ कोलेस्ट्रॉल. लाइक जो HDL अपन कह रहे हैं, दे कंटेन्स लार्जर अमाउंट ऑफ, सपोज करें अपन यहीं पे कंपेयर करने की कोशिश करें तो यहां पे आप जब बनाएंगे, दे कंटेन्स मोर अमाउंट ऑफ कोलेस्ट्रॉल. सो कोलेस्ट्रॉल ज्यादा है, लिपोप्रोटीन्स कम है, तो इसे LDL कह रहे हैं. कोलेस्ट्रॉल कम है, लिपोप्रोटीन ज्यादा है, तो इसको HDL कह रहे हैं. सो द कोलेस्ट्रॉल ट्रांसपोर्टेशन इज इन द फॉर्म ऑफ HDL एंड LDL, and the transportation of cholesterol into the cells, it is a, we can say it is a bulk transport. bulk transport और mechanism क्या कहलाएगी? receptor mediated endocytosis. it is receptor mediated endocytosis process. ठीक है, ये चीज याद रखनी है अपने को. नेक्स्ट एग्जांपल वी कैन टेक ऑफ द आयरन. आयरन ट्रांसपोर्टेशन, अगेन इट इज थ्रू द receptor mediated endocytosis. Suppose in the intestinal human we are having the iron. So iron, it cannot be dumped into the blood in the form of iron. So अगर आप क्या करेंगे, इसको इसी फॉर्म में ब्लड के अंदर डाल देंगे, अगेन देयर इज ए सिमिलर प्रॉब्लम, इट विल गेट क्लम्प्ड, वो एग्रीगेट हो जाएगा. दैट्स द रीजन व्हाई इन द टिश्यू कल्चर एक्सपेरिमेंट जब आप फेरस यूज करते हैं, यू यूटिलाइज दिस Fe along with the EDTA. इस आयरन के साथ में आप EDTA डालते हैं, एंड दिस EDTA इट इज अ चिलेटिंग एजेंट व्हिच डू नॉट अलाउज द आयरन टू गेट एग्रीगेट. जो एग्रीगेट हो गया तो फिर वो कुछ काम नहीं आएगा. तो यूज करना है उसको, तो आपको एक फ्री फॉर्म में रखना पड़ेगा, और फ्री फॉर्म में रखना है तो वहां पे आप देते हैं चिलेटिंग एजेंट्स. सिमिलरली द आयरन, इट कैन नॉट बी डायरेक्टली डंप्ड इनटू द ब्लड. फिर यहां पे क्या करेंगे अपन, हियर द आयरन इट इज एडेड विद ए ट्रांसपोर्टर. दैट ट्रांसपोर्टर इज कॉल्ड एज transferrin. It is transferrin. तो आप क्या करेंगे, आयरन को ट्रांसफेरिन पे ऐड कर देंगे. नाउ इट कैन बी ट्रांसपोर्टेड और डंप्ड इनटू द ब्लड. अब आप इसको ब्लड में डाल सकते हैं. फिर आफ्टर एंटरिंग इनटू द ब्लड, द सेल व्हिच रिक्वायर्स आयरन, इट हैव रिसेप्टर फॉर दिस ट्रांसफेरिन. ठीक है, ट्रांसफेरिन के रिसेप्टर्स उसके पास में मौजूद होंगे, एंड दिस विल receives this transferrin. Okay, again the movement is or the transportation is through the receptor mediated endocytosis. Sorry, जो आपका रिसेप्टर है वो रिकॉग्नाइज करेगा. there is a change in the conformation of receptor which makes the conformational change in the clathrin layer. there is a formation of clathrin coated pit, phagolysosome, and it will release the iron within the सेल के अंदर आयरन को फिर से रिलीज कर दिया जाएगा. नाउ द प्रॉब्लम इज विद इन द सेल, अगेन इट टेंड्स टू गेट एग्रीगेटेड. तो एट दैट पर्टिकुलर कंडीशन वी आर हैविंग अनदर एडेप्टर मॉलिक्यूल, एंड नाउ दिस टाइम द एडेप्टर इज फेरेटिन. तो वहां पे उसको फेरेटिन के साथ में अटैच करके आप रखेंगे. सो द आयरन ट्रांसपोर्टेशन, अगेन इट इज ए रिसेप्टर मीडिएटेड endocytosis, it is transported through the transferrin and the ferritin. So this is all about the specific type of transportation, that is ABC class of transporters and the receptor mediated endocytosis. Thank you.